Greetings, and a good day to each of you. Vaccines are part of everyday medical and pediatric life nowadays, and seem simple in principle. Today, I want to tell you about the decisively less well-known part of the story, the unsung hero, or at very least accomplice, of many vaccine antigens, carrier proteins. First, I will give an overview of the contents of this presentation. In the introduction, I will outline the involved scientists and institutions of the studies. After that, I will tell you more about the main topic of today's presentation, carrier proteins, the concept, how they compare to each other, and which conjugate vaccines are in common use. Then I will provide more details about how exactly the researchers attempted to quantify the effectiveness of different vaccines and compare them. Next, I will provide a list of experiments, display their results, and draw conclusions from what has been gathered. The paper is a review about comparative safety and efficacy of conjugate vaccines against major encapsulated bacterial pathogens. It was published in 2011 in the journal Vaccine, and cited 81 times since in further papers. The review was written by 3 authors, namely Marcus Nuff, Frank Kowalczyk, and Dorothy Kieninger, who has sadly passed away in 2018. All 3 name the fields of medicine, immunology, and microbiology among their subjects, and have published other papers on the topic of vaccination and worked with clinical data. All 3 are associated with the Department of Pediatrics at the Johannes Gutenberg University in Mainz, Germany, and Marcus Nuff, in particular, also with the Children's Hospital of the Dr. Horst Schmidt Clinic in Wiesbaden, Germany. The field of pediatrics is quite important in this context, because almost every study discussed in this review involves trials on children or infants. Child vaccines should both be safe and have as little side effects as possible. One cause of the high infant mortality in the past can be attributed to the lack of vaccines against the important pathogens which we encounter early in life. By 1800, 100,000 had already been vaccinated against smallpox, and then, surprisingly, nothing new happened in the world of vaccines for a long time, until the end of the century, when a whole burst of vaccines was developed. The groundwork had been laid, and yet, childhood mortality was still at 20% in the USA by the turn of the next century. Those that targeted the most common infant diseases came later. And by the 1920s, the development of new vaccines ramped up considerably. For this presentation, diphtheria and tetanus are also of particular importance. Here we can see the fruit of over 100 years of vaccine research, the current German vaccination regime from 2019. By order of the list, these pathogens cause viral diarrhea, from mild to severe, mostly during month 6 until year 2. Bacterial infection with tonic spasms, possible hemolysis and a lethality of 10 to 20% even under intensive care. Bacterial respiratory tract infection which may lead to suffocation in the worst case, and has a lethality ranging between 5 to 10% among children and higher among older adults. Bacterial respiratory tract infection accompanied by a severe cough and a 10% chance of secondary pneumonia. An infant with this infection will need hospital care in about 50% of all cases. Another bacterial infection which can cause pneumonia, but also meningitis and septicemia, and causes long-term complications in up to 20% of all cases. Paralysis, caused by a well-known viral agent, which is asymptotic or mild most cases, but causes long-term weakness in 2/3 of those who experience the symptoms during the acute phase. Another viral infection, which may become a persistent and lead to liver cirrhosis or cancer. A disease caused by Streptococcus pneumoniae, the most prevalent pathogen of bacterial pneumonia, but may also develop otitis media or meningitis in adults, with a case fatality rate of 10 to 30%. An infection caused by the bacterium Neisseria meningitidis, which, as the name suggests, can cause meningitis and leads to sepsis in 1/3 of all cases where the bacterium becomes invasive and is difficult to notice in infants. In septic cases, the lethality is 13%, and long-term effects are also a risk. Another well-known and highly contagious virus. It may also cause otitis media, encephalitis or subsclerotic panencephalitis. Mumps is an infection of the respiratory tract, and, mostly in infants, may also infect the lower parts of the lung or cause infection of the central nervous system. Rubella seldomly causes severe disease, but has a considerable chance of causing miscarriage or development defects in pregnant women. And last on the list, varicella, or chickenpox, is a herpes virus, highly infectious, mostly affects young people, and may also cause pneumonia, encephalitis, and cognitive disability in unborn children. This was quite a long list, but I found it important to outline the reasons why it is important to vaccinate children at a young age. These are the pathogens which will be important for the later parts of this presentation. Let's begin with the first group, tetanus, or rather, Clostridium tetani. It's gram-positive, obligate anaerobe, motile bacterium, which can also form spores that are very resistant to heat and chemical disinfectants, and ubiquitously found in the soil. Infections in humans may occur wherever the skin barrier is broken, so a single tiny scratch while playing outside may be enough. The toxin, here shown at the bottom with its secondary structures colored, is cleaved and activated by a protease. This means inhibitory neurotransmitters are no longer released, which leads to cramps and muscle spasms. And second on the list is Corynebacterium diphtheriae. It's also gram-positive, but not motile, and facultatively anaerobic. Unlike Clostridium, it can't form spores. Its toxin is encoded on a lytic phage. It acts by modifying by binding to host cells, entering, and then catalyzing the inactivation of the host ribosomal elongation factor EF2, which stops protein synthesis completely. Last in the first set, Bordetella pertussis, gram-negative, and it is aerobe, preferring to colonize the respiratory tract. Also, it has a whole host of virulence factors, among them fimbria, multiple toxins, hemolysin, immune response modulators, and of course the pertussis toxin, which inhibits G-protein-coupled receptors after entering. The toxin is of the type AB5, which means that the binding domain is a pentamer. And now we move on to the second set. Haemophilus influenzae is a gram-negative, immotile, human-specific bacterium which mostly infects young children. There exist strains with and without a capsule, or typable and non-typable. Haemophilus influenza with capsule can be assigned to the serogroups A through F, and type B, the most common one, produces a polyribosyl ribitol phosphate, PRP, capsule, which is a major virulence factor. Also, it has an IgA protease, which inhibits agglutination and clearance at mucous membranes, and from the age prevalence, we can see that the highest incidence is among infants. Streptococcus pneumoniae is a gram-positive diplococcus, which is also non-motile, and has a capsule, consisting of complex polygalactose carbohydrates. There are many serogroups, but most cases of disease are caused by a few specific ones. An invasive pneumonia is mostly found among young children or the aged population. Here are again the cases per 100,000 members of the population per year. Neisseria meningitidis, causative agent of meningococcal meningitis, also occurs as diplococci, but is gram-negative. As the other two shown here, it also has a capsule, and there are 12 serotypes, among which 5 are associated with disease the most. And here we can see the age prevalence of invasive Neisseria in Italy from a few years ago. As with Haemophilus, it predominantly affects young children. Here in the overview, I want to highlight a very important factor for these pathogens, and by extension also the development of their respective vaccines. And that is the fact that the three all have a capsule, which makes the host immune response much more difficult. The polysaccharide masks the bacterial surface proteins, and is also highly variable, which means that each pathogen variant necessitates its own adaptive immune response and refinement. And most importantly, in the context of vaccines, polysaccharides can't generate immune memory. Which leads us to the concept of conjugate vaccines. Let's compare the vaccine creation dates of the 6 bacterial pathogens I've just presented. The first 3 were invented much earlier. I assume at least part of the reason was that it was difficult to create a vaccine which develops a robust memory response, and indeed, the first conjugate vaccine was created as late as 1985. The reason for the lack of a memory response is as follows. Without a protein component, an antigen can't be attached to a MHC molecule, which means it can't be presented by APCs. Only B cells with a matching B cell receptor may interact with the polysaccharide directly and produce antibodies. However, without the help of T cells, there is no B cell maturation, which means that no immune memory against the antigen will be developed, and the produced antibodies will not improve in their affinity during the infection. To circumvent this, vaccine researchers did a very smart thing. They artificially attached a protein component to the polysaccharide, which meant that the immune reaction could now be T-cell dependent. This image sums it up very nicely. Without conjugate, the B cells do not receive the co-stimulation and proliferation signal from T helper cells in the lymph node, and die eventually. With, they may affinity maturate, and form long-lived memory cells. As conjugate, they used, among others, proteins which were very well known, and whose effects in the human population were already well studied. Some of which are old friends, like the diphtheria toxoid, or in other words, the diphtheria vaccine from 1923, and the tetanus toxoid vaccine from 1924. Then there's also another protein from diphtheria, called diphtheria non-toxic cross-reactive material, or CRM. Its advantage is that unlike DT or TT, it does not need to be detoxified through chemical means before injection. Next there's PD, which stands for Haemophilus influenza protein D, a conserved surface lipoprotein. And lastly, the outer membrane protein, OMP, from Neisseria meningitidis. Most of these have different molecular weights and shapes. It was the objective of the review to compare the effects and results of different vaccination formulations in clinical studies, mostly performed on children. So that's where we will move next. To compare results, we must first define a quantity which measures the success or failure of a vaccine. For this purpose, the review mostly used the SBA titer, which stands for serum bactericidal antibody, a measure of the pathogen-specific functional antibodies that are necessary for protection against infection. A serial dilution is performed on blood samples. If there are specific antibodies against the pathogen, they will be attacked and lysed by complement. After the incubation phase, the viability of the remaining will be checked, either by spreading the remainders on a culture plate, or with chromophores which change their absorption in the presence of live bacteria. In the studies, a protective immune response was quoted once neutralization was achieved at a dilution of 1 to 8 or lower. Then, there was also the quantity of AEs, or adverse events. This does not quantify a protective immune response, but rather the number of unpleasant or threatening complications caused by the vaccination within the study population. For example, these could be local reactions at the injection site such as redness, pain and swelling, immediately after vaccination or the day after, but also hypersensitivity, febrile seizures, and in the worst cases, anaphylaxis. To understand which compounds were injected, it's also important to understand the nomenclature of vaccines. Here's a vaccine called MenACWY-CRM, brand name Memvia, which is a vaccine against meningococcal meningitis. ACWY means that it protects against the serogroups A, C, W, and Y, and the polysaccharides are coupled to the CRM protein. For these three it's similar. PNC stands for pneumococcal, so Streptococcus pneumoniae, and the number for the serotypes. Since most infant vaccination regimes plan multiple vaccinations in a short time, some vaccines are combined for the sake of convenience. In these cases it becomes a little more complicated. In one of the studies, one group was injected PNC4TT, so a quadrivalent tetanus toxoid conjugated Streptococcus pneumoniae, together with DTP-IPVPR-PTT. DTP stands for three different vaccines, namely diphtheria, tetanus and pertussis antigen. IPV is an activated poliovirus, and PRP for the Haemophilus influenza type B polyribosyl ribitylphosphate, conjugated to the tetanus toxoid. A more definite name for DTP is DTAP or DTWP, which stands for diphtheria, tetanus, acellular pertussis or diphtheria, tetanus, whole cell pertussis respectively. The acellular variant is the more recent invention, and consists of different antigens depending on the manufacturer. And now that we've covered the basics and theoretical background, we can move over to assessing and interpreting the results of the studies along with the authors. In total, 18 comparisons have been made, and of those, the overwhelming majority, 15, focused on the robustness of the immune response. Only 3 compared safety profiles or numbers of AEs. In the individual studies, they mostly tested different vaccine combinations. Only in one case they compared the effects of a vaccine to a non-vaccinated control group. Among those were also a few new experimental vaccines, and where the information was given, those took place as Phase 2 or Phase 3 trials. Here I have attempted to create an overview table of the individual comparisons in the order in which they appeared in the review. Now I will go over the experiments, outline their scope, and their conclusions. Since there are so many, I'll try to add the conclusion of each comparison to this list, so that we can still keep an overview. I will now begin with the comparisons of vaccines against meningococcal disease. In this study, they compared the safety. Both received a quadrivalent meningococcal vaccine, of which one was conjugated with CRM and the other with the DT carrier protein. From the results we can gather that the DT conjugate more often led to pain as a reaction, but the difference is not large. Since CRM appears to be better than DT, at least under the conditions of this study, I'll enter this information with a larger sign. Please note that this is one of the few studies that was not performed with infants, but rather adolescents. In the next study, they compared the safety of the quadrivalent meningococcal vaccine, conjugated to TT, to the licensed monovalent vaccine conjugated to CRM. They found a similar incidence of local and systemic adverse events, so at least in terms of safety, the experimental quadrivalent vaccine appears to be comparable to the licensed one, and should offer a broader protection. In this study, they compared the serum antibody response of 3 different commercial vaccines when given at 2 doses 2 and 3 months versus 3 doses at 2, 3 and 4 months. Shown is the percentage of infants which reached the SBA larger or equal than 1/8. As we can see, all 3 preparations resulted in very good protection after the last dose. However, NeistVac-C already reaches this SBA level after the first dose in 97% of infants, whereas the other 2 only reach 53% and 80% respectively before the booster vaccination, so it can be considered superior to the others in terms of protection at the start. On the other hand, Menjoget reaches 100% protection coverage whereas NeistVac-C misses out on 2%, so it's difficult to say which vaccine offers better protection and saves more children from disease overall. I'd say NeistVac-C, which is a MEN-CTT conjugate, is the best choice among these 3. The next study confirms the finding of the previous one. Again, MEN-CTT and MEN-CCRM were compared, and it was found that MEN-CTT already offered very good protection after the first dose. In this study, they tested the immunogenicity of MEN-ACWY conjugated to 3 different proteins in very young infants in particular, at 2 months of age, and found that only the CRM conjugate offered, quote, adequate immunogenicity. Here they compared TT and CRM and found that TT led to more antibodies than the serum. Since the infants also received a HIPTT vaccine, the authors assumed that the already existent memory against TT enhanced the immune response against the MEN-C vaccine since it used the same carrier protein. In a similar setup, another study found that after the 3rd dose of a MEN-CTT vaccine, compared with the CRM control, the antibodies against MEN-C were lower, while the antibodies against the Haemophilus influenza antigen, whose vaccine was given at about the same time, was higher. The authors of the review assume that TT can have either a positive or a negative immune interference effect, depending on the circumstances and other administered vaccines. After the vaccines against meningococcal infection, we have now arrived at the part about Haemophilus influenza vaccines. In this first study, they compared the incidence of adverse events after vaccination with Haemophilus influenza antigen conjugated to different carrier proteins, and found that the TT conjugate caused the most cases of crying or fever, while DT and CRM had the fewest cases. Here, they compared the immunogenicity. From the development of the antibody titer in blood after the 2nd and 3rd dose, we can once more see that it's hard to determine a vaccine that's objectively the best in every case. I would say that the TT conjugate is better than the other 2 since it reaches the highest percentage of infants with a titer above 1.0 microgram per milliliter, but that's debatable. This study is similar in its setup. However, they also tested the DT conjugate. Somehow the percentages were smaller, but TT once more appeared to be the best. Now we're finished with the HIP vaccines, and moving over to the pneumococcal vaccine comparisons. Here they have compared the generation of anti-pneumococcal antibodies after injection of a heptavalent pneumococcal vaccine compared to a meningitis control, and, as might be expected, found that after vaccination with the pneumococcal vaccine, there are also much more anti-pneumococcal antibodies to be found. Someone else did a similar study, only with an undecavalent pneumococcal vaccine instead of a heptavalent, this time conjugated to PD, with similar results. Also, they checked the protection of the vaccine against otitis media, and found that the pneumococcal vaccine provided better protection, as might be expected, even though those without this vaccine were not entirely defenseless either. In another study, They compared the immunogenicity of quadrivalent pneumococcal vaccines either conjugated with TT or DT, and found that both produced antibodies, however DT more so than TT. Also, the fourth booster vaccination of DT at 12 months lead to a greater increase in antibody titer than TT. In this study, they injected quadrivalent pneumococcal vaccine, however, they measured the antibodies against Haemophilus influenza antigen, and found that the titer was lower when a vaccine was applied. Also, it was lower for the TT conjugated compared to the DT vaccine. The authors theorized that this was caused by TT carrier protein overload and immune interference, so a situation where the immune system is already working at maximum capacity. And more or different antigens with similar components decrease the antibody output, although the titer was still high enough to be protective. A similar study was performed where the mutual carrier protein was CRM instead of TT. Both groups received a vaccine which included HIB-CRM, and one a vaccine with nanavalent PNC and MenC, which adds up to a total CRM content of 64 microg, while the control only received 25 microg in total. Unlike the previous comparison, this time, a positive effect of additional carrier protein could be found, and the total antibody titer of anti-HIB, which was not a content of the sample vaccine, increased. Here, the authors suspect that CRM acted like an adjuvant, and thus boosted overall immune response. This one is similar again, however, this time the concomitant carrier protein was TT, and the sample vaccine contained CRM. This time, the antibody response was decreased by the additional CRM protein, and not only the antibodies against MenC, but also the other antigens, HIB and diphtheria, decreased. The authors propose a case of protein overload, just as in comparison 12. The last two comparisons were made between whole cell and acellular pertussis vaccine. In this study, they explored the influence of those two forms of pertussis vaccine on the antibody response to other concomitant vaccines, and found that DDAP acted as inhibitor on the response against those pneumococcal antigens that were conjugated to TT while not significantly changing the level of other antibodies. And finally, the last comparison of this presentation. Here, they measured the HIB and MenC antibody titer after either DDAP or DTWP and either MenC-TT or CRM. As a result, they found that DTWP together with MenC-TT increased the MenC antibodies, or in other words, DTWP improved MenC-TT response, while DDAP decreased it. Also, MenC-TT improved the anti-HIB response more strongly than MenC-CRM. Unlike the previous study with PNC, here, DTAR increases the TT conjugated antibody response. This concludes the presentation of the different studies. As you have seen, there are many varying factors we have to take into consideration, not just the vaccines applied during the study, but also different concomitant vaccinations with different carrier proteins, as well as different time frames, dosages, age groups and study sizes, and sometimes even contradictory results. And since we are dealing with immunity, even the different environmental exposures of different populations might influence immune reaction in unforeseen ways. Here, I will just try to summarize the findings of the studies. We have seen ones that observed the safety of vaccines by counting adverse events, gauging the immune response of vaccines by their own, either by SBA titer or concentration of antibodies in milligram per milliliter serum, or elucidating the cross-reaction between different vaccines and carrier proteins. During these trials, we have seen that the tetanus toxoid seems to perform best in most studies, except in a safety study where it performed worst. Maybe TT acts as a stronger adjuvant than the other three options, and this way, leads to both more adverse events and a stronger antibody response by stimulating the immune system strongly. Also, one vaccine with TT may decrease the antibody response against another with the same carrier protein, or be either increased or decreased itself by addition of acellular pertussis vaccine. We must not forget that we are trying to determine the reaction of a highly complex system of interlocking activatory and inhibitory processes in the human immune system. In reality, it seems to be more like a spider web. If you tug on one of its strands, everything moves. In this slide, the authors have summarized the different vaccine interactions they found in their sources. The interactions had varying effects, sometimes increasing and sometimes decreasing the antibody response, and it was difficult to determine an underlying theme. Lastly, I want to summarize the presentation and the review in a few sentences. Vaccines are complicated. There are many different formulations and multiple carrier protein choices. They all can interact to some extent, And not just the content of the current vaccine matters, but also those of previous vaccines. However, all established vaccines are maybe not perfect, but good enough. Their introduction has severely reduced child mortality and long-term disability in most parts of the world. Some vaccines and combination may be better or worse, but most are already quite good. Even so, that does not mean we should not try to improve what is already a rather good system. When we are applying a preventative medical procedure to millions of children, even small percentages can make a difference on thousands of lives. And this concludes my presentation of the topic of carrier proteins and their interactions. And these were my sources. Thank you for listening, and I hope you've learned something interesting today.